Okay, before we get started moving into microtubules and stuff, I got a proposition I want to run by you, okay? It's been brought to my attention by some people in here, by looking at the calendar and talking to one of my esteemed colleagues who teaches another course this fall, who does so with a very animated Irish accent, and makes me look like I'm a crawler as far as speed. Anyway, here's what I want to run by you guys. What would you think about having the next exam pushed another week? Instead of doing here, no, hear me out. Let's make cell biology great again. Here's why. Okay, here's why. It would not put more material on the exam. It would still be cytoskeleton and signaling that will be just starting on Friday. We're running about a class behind, and that's by we meaning me, and that's because, according to my colleague from the Emerald Island, you're slowing down, dude. The ATP is just not there. So, yeah, I've kind of slowed it down a little bit. And what I don't want to have happen is I don't want to be having a substantive lecture finishing up the last bit of signaling on Monday, November 8th or 7th, and then and then we got this election day we all have to get through. Most of us are either going to be hungover because we're depressed, or we're going to be celebrating on that Wednesday, on the 9th. So I am perfectly happy to push the exam to the following week, the 16th. It's the week before Thanksgiving, so there's no things there. So I'm just I haven't decided yet what to do. I just want to if you show of hands, just kind of show me how many would want to do it. Okay, and how many don't? Okay. So that's about 50-50, okay. So, looks like, if that's the case, I'll probably keep it the same. Hey, it's 50-50, sorry. It's rigged. The media's against me, you guys so I can't do anything, okay? It's all rigged. You're going to hear about it tonight for 2 hours. Okay, so I'll just keep it the same. If it means that I have to speed up now, I will. But, so anyway, we'll just keep it the same and see how it goes. I just wanted to run it out there. But I know that it's one of these things where I know that if in any given week, you guys are being slammed with a set of exams, and if you push it, you're just moving it into another hassle. So we'll just keep it where it is. Okay, I just wanted to throw that out there. Blame Condren. I didn't realize I had slowed down that much. Okay. All right, into microtubules. All right, I don't have time to talk about it. We're done. Okay, microtubules. Far more dynamic than microfilaments. Okay, if there's one thing to grab right at the beginning looking at this, microtubules are at least 1,000 times more dynamic than their microfilament counterparts. Even in a cell that is sitting there doing absolutely nothing. It's not crawling, it's not moving, it's not changing its shape, it's not dividing. Microtubules, with the rare exception of neurons, and maybe I'll get Condren and drag him in here, and he can tell you about that. Neurons are one of the very, very few cells where microtubules are infinitely stable. The lifespan of microtubules inside of neurons is literally the lifespan of the cell, okay? But in most other cells, they're incredibly unstable. And the paradox that you're faced with looking at microtubules is a phenomenon which is known as dynamic instability. And what dynamic instability has to explain is the following. Why is it in a given cell, under identical conditions, you've got two microtubules right next to each other. One of them is growing and the other one is shrinking at exactly the same moment in time. And that's what we have to try and come to grips with explaining when we're looking at microtubules. So the instability is going to play a major role when we get to mitosis, because making and functioning of the mitotic spindle requires microtubules to undergo significant changes in their dynamic properties. We'll get to that in mitosis. So in this section, we're just going to be talking about what underlies the abilities of microtubules to grow or shrink. Okay? The next part of it that we're going to deal with, though, are how motor proteins that are involved in carrying all the vesicles that we talked about in the last section, how do motor proteins function using microtubules as a highway infrastructure? And how does it solve the problem of directional movement of vesicles in a cell? Not so much where they go in terms of how do I know I'm supposed to fuse at this target membrane or a different one. We went through that. This is the actual mechanics of how they get there. Okay? The structures are large. They're much larger than microfilaments, which are little pieces of string that are about 8 nanometers in diameter. These are literally tubes. They're hollow on the inside. All sorts of lunatic fringe ideas about what happens on the inside of a microtubule. Some people think they're the seat of consciousness. Memory is imprinted on whatever goes through the inside of microtubules. We're not going to get into that. We're more concerned about what do you do on the outside. Okay? You can actually see they're rather large, about 25 nanometers. And the other thing that you can notice, if you take a look at this picture up here, this immunofluorescence image of a cell stained with tubulin, is that in a non-neuronal cell, you can trace all the microtubules in that cell back to a single origin. And it's called the microtubule organizing center, or the MTOC, also known as the centrosome. And as you're going to see, within the centrosome are a pair of centrioles. And that is where all the microtubules originate from, in non-neuronal cells. Neurons have their own way of dealing with this. That's very, very different than microfilaments. Microfilaments spring up all over the cell. And what you're going to see is, when we trace all the microtubules in a non-neuronal cell back to the MTOC, that's where all the minus ends are embedded. So the neat thing with microtubule biology is that in rare exceptions, all the action takes place at the plus end. We don't have to worry about the minus ends. They're basically invisible. Okay? Now, microtubules are a little bit more complicated than microfilaments. Microfilaments are comprised solely of the protein actin. So you just polymerize G-actin to F-actin, you make a microfilament. Actin, as you guys know, is an ATP binding protein. And it's also an ATPase. So it has the ability to hydrolyze ATP to ADP. Tubulin is a heterodimer. Contains two separate polypeptides, each encoded by their own gene, alpha tubulin and beta tubulin. There's many, many, many different isoforms of alpha tubulin and beta tubulin that can be coming together to generate microtubules with different functions and different cell types. We're not going to worry too much about that. You never find free alpha or free beta in a cell. As soon as you make either one of those polypeptides, they immediately come together. To separate them, you have to take this complex, this heterodimer, and you'd have to boil it in like 1 molar mercaptoethanol, 6 molar urea, and you might be able to pry them apart. So these are the two most stable proteins in terms of their interaction that you are ever going to find in a cell. So because of that, we actually refer to this heterodimer as tubulin. So when we talk about tubulin, that's what we're referring to, an alpha-beta dimer. And it's just because you're never going to find alphas free or betas free. Tubulin is it's a G protein. So that's already telling you there which nucleotide's involved. The only one that matters is beta. The GTP that's bound to alpha is non-exchangeable, and it's almost, you can consider it non-hydrolyzable. It's locked in there. It's inaccessible. So as soon as you make an alpha tubulin, it binds GTP, and it's got GTP bound to it for the rest of its life. Okay? So GTP bound to alpha, don't worry about it. Here's the important one, beta. And the reason is the GTP on beta can be hydrolyzed to GDP, and that's going to be the major determinant of microtubule stability. Microtubule dynamics are all going to be driven by rates of GTP hydrolysis on beta tubulin. So that GTP is referred to as exchangeable. And the reason is, once it's hydrolyzed from GTP to GDP, the GDP is exchanged. We don't need a gap here. And the reason is, if you take a look at the structure, it's exposed. So as soon as beta tubulin hydrolyzes GTP to GDP, the GDP can diffuse away, and we can replace it with a fresh GTP. Okay? That GTPase activity is intrinsic to tubulin. You can inhibit it, you can incubate it with non-hydrolyzable GTP. The other thing that you can do is that there are drugs that can affect the GTPase activity of tubulin. And the one that we'll get to in detail down the road when we talk about mitosis is the anti-mitotic chemotherapeutic drug known as Taxol, or paclitaxel. There's the structure of it. It is incredibly cytotoxic. And the reason is that as you're going to see, it inhibits GTP hydrolysis by beta tubulin. That means that if you treat a cell with Taxol, you have microtubules that cannot depolymerize. And that's going to stop mitosis. Specifically, it's going to stop it during anaphase. So cells are not going to be able to segregate their chromosomes. You're going to get missegregation of chromosomes during mitosis, and the cells are going to die. So Taxol is a very potent chemotherapeutic drug because it's going to go after and interfere with mitosis in rapidly proliferating tumor cells. The problem is it's not specific. So it's going to prevent GTP hydrolysis by beta tubulin in any cell that it gets into. So the problem with Taxol, as some of you may know from individuals that have been treated, for example, with highly aggressive ovarian or breast cancer, you have all these horrific side effects. hair loss, anemia. The reason is the Taxol will stop mitosis in any and all proliferative cells. So as you're going to see when we get to cancer, the way to utilize the power of this chemotherapeutic drug is to try and alter it in such a way with a delivery device called a smart bomb. And that is to take a drug like Taxol, conjugate it to an antibody, that the antibody can then recognize a protein on the surface of a tumor cell, and basically that's how you could deliver this chemotherapeutic drug specifically to just a cancer cell. And we'll talk about some applications of that in a few weeks. Okay? All right, so this is the basic building block. You can see it's got polarity. You've got beta over here on one side, alpha on the other. And just like microfilaments, we're going to assemble microtubules with a polarity in the structure. What we do is that the assembly starts by taking tubulin, and you can see it's referred up here as a dimer. Okay? So in essence, what you're going to see is that they're all arranged head to tail. And what they assemble into is you first start polymerizing tubulin into little linear chains that are referred to as protofilaments. Once the protofilaments get large enough, typically about a dozen tubulins, the protofilaments come together and they will spontaneously assemble and make a microtubule. All right? The most common microtubules that you find in our cells are referred to as singlets. Singlet microtubules have 13 protofilaments. So what happens is, once you elongate these individual protofilaments, again to about a dozen in length, they come together and they make the microtubule. Once you have this now, what you're doing is that if this microtubule was growing out of the slide, sort of coming out at you, you would be adding tubulins to all of those protofilaments that you find in that microtubule. Okay? What you're going to find, all the action takes place at the plus end. The plus end has the exposed beta tubulin. The alpha tubulin is down at the minus end. And what you're going to see is in the centrosome, we're actually going to wrap another specific form of tubulin on that alpha called gamma tubulin. And that, in essence, is what's going to make the minus ends invisible. They're not going to be relevant to any of the dynamics that we're going to be going through. Okay? So what it means is they're all lined up like this. You can see there's a seam, and the seam is in between each protofilament. And you're going to see that there are proteins that are going to bind to the seams that are going to influence the structures of microtubules that we can form. Okay? Now, like I said, singlets are the most prevalent form of microtubules within our cells. But you can have certain instances in ciliated or flagellated cells, for example, like a sperm cell, where instead of finding singlets, you find doublets. And doublets are just a pair of microtubules that are fused together, and they share 3 protofilaments in common. Okay? Now, if you take a look at a cell, again, a typical non-neuronal cell, and you trace all those singlet microtubules all the way back, as far as you can go, into the centrosome, what's in the centrosome are triplet microtubules. Okay? So these are what you find within the centrosome, and then what happens is as we come out of the centrosome, only one of these fused rings will actually continue to grow. Okay? So they start off as triplets, but then they just spew out singlets that emerge from that. The other thing to note here is that just like with actin, remember with actin, when you make a microfilament, the ATP hydrolyzes after the actin goes into the microfilament. GTP on beta tubulin hydrolyzes after it goes into a microtubule. And you might say, well, what's causing that? Well, as you're going to see, there's a gap involved here. And the gap is going to be the alpha tubulin on the next one that adds in. So this little guy right here, this tubulin that's right at the plus end of a microtubule, that beta will initially have GTP. But as soon as we take the next tubulin and we add it on to that, what you're going to see is the alpha coming up next to that beta acts as a gap and causes it to hydrolyze GTP. So everything's built into tubulin. We don't have to worry about so many extra proteins like we did with microfilaments. So in that way, it makes it actually a little bit simpler. Here's where they all originate from, the centrosome. And within the centrosome, we've got a pair of centrioles. We're going to talk about these when you get to mitosis. These are perhaps one of the most poorly understood structures in our cells today. They duplicate on their own. Centrioles duplicate during S phase, just like our chromosomes do during DNA replication. As you're going to see, they duplicate semi-conservatively. Interestingly, we refer to centrioles as mothers and daughters. This mother-daughter pair will separate when the cell goes into S phase to replicate its DNA. And this mother will now promote the formation of a new daughter. This daughter will promote the formation of another daughter. It sounds kind of sexist, but we basically have to deal with a feminist view here of how the centrioles are designated. But they replicate semi-conservatively. There's no DNA involved here. The process by which this takes place is very poorly understood. It's important because where the centrosomes migrate determines the axis of cell division. Your chromosomes have nothing to do with that. It's where the centrosomes migrate after they duplicate that determines if the cell is going to divide horizontally or if it's going to divide vertically. We have very, very little understanding of how centrosomes know where they are supposed to go when the cell is going to divide. Okay? So, the key thing that we're looking at right now is that you notice they're also, here's another funky thing, they're always perpendicular to each other. One faces up and down, the other faces left, right. No one has a clue why they do that either. You can see that if we flip this guy towards you, this is the arrangement of those triplet microtubules within that centriole. So in a non-neuronal cell, half of all the microtubules in that cell are coming from this centriole, and the other half are coming from this one. If you work your way down to the very bottom of that structure, so here's a microtubule out here. If we trace it all the way down to the M-top, there's the last alpha. What we add right to that alpha then is a very unique form of tubulin that's called gamma. Gamma is only found in centrioles. We don't find it anywhere else. And then gamma is also associated with a group of other proteins that are still very poorly characterized, and it's called the gamma-tubulin ring complex, or gamma-TERP. The important thing for our perspective is it means that the minus ends of microtubules are invisible. We don't have to worry about them. All we have to be concerned with is everything that's going to be going on at the plus end. Okay? So it makes it a lot easier to go through the dynamics when we're only having to fool around with what's going on at the plus end. Okay? One more thing. If you take a look at these cells here, one of the consequences of this is that with all the minus ends embedded in the M-top, all the microtubules that are coming out towards the plasma membrane, they've got polarity. Think of them as one-way roads, because what it means is all of the microtubules in that cell are oriented with their minus ends here, and their plus ends are out there. This is why we're going to need directional motors to carry things on the microtubules. Because if you want to move from the center of the cell towards the plasma membrane, we're moving towards the plus end. That means we have to use a plus end motor. If we're bringing something into the cell, like LDL receptors bound to LDL particles, if we're endocytosing those and bringing them towards the lysosome, we need to use a minus end motor. Okay? So the motor proteins that are going to carry vesicles on the microtubules are directional. They can only move towards the plus end or they can move towards the minus end, but they're not bidirectional. Okay? So this polarity that you're seeing right at the outset lays the groundwork for understanding why we have to have different types of motor proteins to use the microtubules to transport things. Here's another funky thing about microtubules. It's just sort of throwing this up there as a little curiosity. Let me go back to this slide here. See these cells? They're happily existing there at 37 degrees. If you take these cells and put them in an ice bath so you cool them down to 4 degrees, the microtubules disappear. They depolymerize spontaneously. So one of the things that's different between microtubules and microfilaments is that microtubule structures are temperature dependent. So if you take cells or purified microtubules and you have them at 37 degrees, cool them down to 4 degrees. Take your cells or take your purified microtubules and put them in an ice bucket and walk away for a few minutes and come back. The microtubules are gone. They've all depolymerized into free tubulin. And again, by tubulin, we're talking about alpha-beta dimers. Come back down. If you take them now, take that same solution of purified tubulin or cells that are now at 4 degrees and warm them back up to 37, they regenerate. So you can get your microtubules to reform again at 37 degrees. It's reversible. You can take advantage of this to purify microtubules from cells. And the reason is you can use this to crystallize them. So if you take a cell-free extract, incubate it at 4 degrees, the microtubules will all depolymerize to that free tubulin. Take that extract now and put it at 37 degrees. The microtubules will all precipitate out a solution. You can literally centrifuge them out. So you can literally crystallize and purify by low-speed centrifugation microtubules out of cells just by cycling extracts between 4 degrees and 37 degrees. It's a very, very simple way to purify pure microtubules. Okay? Okay. Critical concentration. We got to deal with that again. It's the same thing you saw with microfilaments. And that is if you start with purified tubulin and you look at the conversion of tubulin to microtubules, you have to get above the critical concentration. But here's the cool thing. You don't have to worry about the minus end. Minus end's invisible. We don't have to deal with it. All we have to look at is the critical concentration for the plus end. Because these guys, nothing's going to happen there inside of a cell. They're all socked away in the M-top. You don't see them. So the critical concentration to add tubulin onto the plus end of a microtubule is 0.03 micromolar. It's pretty low. Here's the cellular concentration of tubulin. It's about 10 to 20 micromolar. So one of the things that you guys can see right off the bat, the intracellular concentration of tubulin far exceeds the critical concentration. So if you took a look at that, you would say, all of our tubulin in our cells ought to be polymerized. There shouldn't be any free tubulin. But here's where dynamic instability becomes an important factor. And what you're going to see is, the more you grow this microtubule, the less stable it's going to become. And that's going to be due to GTP hydrolysis. So microtubules, as we make them, are designed to depolymerize and fall apart very, very rapidly in response to GTP hydrolysis. And the cells are going to use that mechanism very effectively during mitosis to separate your chromosomes very rapidly. Okay? This is what dynamic stability is all about. You can do this in vitro with purified microtubules. You can do it in a cell. Okay? If you take a look down here, here's a series of time-lapse images over about a 3-minute interval. And you're looking at 3 fluorescently stained microtubules. You just pick them out, A, B, and C. Okay? And notice that during this time frame, A grows, B stays the same size, but then you notice by the time you get out here after 3 minutes, A has shrunk. Okay? So the point is, these 3 microtubules are in the same cell at the same time. You can have one over here that's not doing anything, one over here is growing, and then the next minute it shrinks. So if you went through and you looked at every single individual microtubule in a cell, this is all at 37 degrees now. What you notice is microtubules are constantly growing and shrinking. But notice the rates of these 2 processes are not the same. You can determine that by looking at the slope of the line. So here's a microtubule here. And you can see that it starts off at a certain length. This would be like 15 microns. It undergoes assembly. And you notice that in this particular cycle, it made it all the way up to 35 microns. Now it undergoes a process called catastrophe. It's only a catastrophe for that individual microtubule. It's not a catastrophe for the cell. What happens at catastrophe is that microtubule that was growing happily 1 minute, the next instant it's depolymerizing very rapidly. And here's where you can see the difference in the kinetics. Notice that the slope of a microtubule that's undergoing disassembly or depolymerization is steeper than the slope of when they're assembling. So once a microtubule starts shrinking, it goes very quickly. But also notice it's not completely gone. It's going to shrink down to a certain length. And in this case, that particular microtubule shrunk more than it was at its beginning. But it undergoes a process called rescue. And that is now, at some point, depolymerization stops, and now we start growing it again. That's rescue. Now you notice, though, this microtubule originally started off up here, grew up to 35 microns. The next round of assembly, it may only get up to 20, and then it shrinks again. But you notice it didn't shrink as far as it did the first time. Now it will grow even longer. So here's the point. You cannot predict for a given microtubule how far is it going to grow before it undergoes another catastrophe. You can't predict when it undergoes a catastrophe how far it's going to shrink before it's rescued. Okay? Your microtubules are doing this all the time in a non-neuronal cell. This is the life of a microtubule. It's never just sitting there doing nothing. They are constantly either growing or they're always shrinking. Intrinsically, that's why they are so dynamic. You look at that cell outwardly and you see nothing going on. But you follow the microtubules like this, and they are constantly changing. Always growing, always shrinking. Okay? So this is what accounts for it. This is a model that was worked out a number of years ago by Mitchison and Kirshner that's called the dynamic instability model. And what it says is, how do we explain a microtubule growing 1 minute and it shrinks the next? And what it relies on is GTP hydrolysis. To a certain extent, it relies on the concentration as well. So let's say here, we've got a microtubule that's in the process of growing. Okay? If you take a look at it under the electron microscope, you can basically see there's the ends. We've got some tubulin here that's being added on. Okay? Remember that what happens is, every time you add a new tubulin to that microtubule, the one that was there, this guy here, as soon as we add this tubulin on, that guy hydrolyzes its GTP to GDP. So what it means is the following. The plus end of a microtubule has what's called a GTP cap. And that means that the very, very last beta tubulin that just went onto that microtubule still has GTP. But as soon as you add a new tubulin onto it, that GTP gets converted to GDP. So what it means is this, guys. As you're growing a microtubule, so as this guy is polymerizing, all of the beta tubulins within the body of the microtubule have GDP instead of GTP. That's changing the structure of the microtubule. And what it does is the following. The protofilaments, when they've got GTP bound at beta tubulin, are nice and linear. But as they get converted to GDP, the protofilaments adopt a curvature. So as you hydrolyze GTP to GDP, these protofilaments start curling away from each other. The easiest way to envision it is it's like the skin on a banana as you're peeling it. Okay? So what it means is this. All of those 13 protofilaments in a singlet microtubule, on that microtubule, are desperately trying to get away from each other. And all it takes to cause the entire structure to fall apart is that there's a pause or a hesitation in adding the next tubulin onto the plus end. And the reason is, if you delay that, then that beta tubulin at the very plus end will hydrolyze from GTP to GDP all by itself. Remember, it's a GTPase. So all that means is the only thing that is holding a microtubule together is the fact that the very terminal beta tubulin at the plus end has GTP bound to it. Convert that beta tubulin to GDP, which is what happens if you don't add a new one with a new GTP, the whole structure falls apart. It literally adopts this split-in sort of structure. In this electron micrograph, you can basically see all the individual protofilaments are splaying away from each other. And once this process starts, the entire microtubule starts to depolymerize until it's rescued. So what happens is, as soon as these protofilaments start splaying apart, the tubulin starts falling off and depolymerizing, and this goes very, very quickly. The rate of doing this is much more rapid than the rate of assembly. So that microtubule, once it starts depolymerizing, it goes very, very quickly until it's rescued. We have to rescue it to get that microtubule to grow again. Okay? It has to do with GTP hydrolysis. So, can you guys see why Taxol prevents this from happening? If you treat cells with Taxol, that's going to inhibit GTP hydrolysis. That's why Taxol is going to make your microtubules very stable. They can't undergo this depolymerization. If they can't depolymerize, you can't undergo anaphase when you get to mitosis. We're going to take advantage of this. We're going to use Taxol to stabilize microtubules in vitro so we can study vesicle trafficking. So when we want to study how we can move vesicles along a microtubule, we have to take microtubules and keep them stable. And the only way we can keep them stable in vitro is to incubate them with Taxol. And that prevents them from hydrolyzing GTP to GDP, and then that allows the structures to remain stable. Okay? Here's the guy who's causing it to happen. Alpha-tubulin. Okay? So here we've got the protofilament at a microtubule. Okay? You just have to look at one of them. The same thing's happening in all 13. So here's the protofilament that comes down. Here's the tubulin that just got added. It still has GTP bound to it. It hasn't had time to hydrolyze it yet. But now, here comes the next guy to add on to the plus end. So we add this on, and now our new plus end is this guy. It still has a GTP cap. But here's where alpha does its job as a gap. As soon as this alpha comes in, binds to what used to be the plus end beta, it causes it to hydrolyze the GTP immediately. So can you guys see that you're building a more and more unstable structure? As this microtubule gets longer and longer and longer, it becomes progressively less stable. And the reason is you're converting all of those beta tubulins along the entire length of that microtubule to GDP. The protofilaments are starting to come apart. And it's only going to remain stable as long as we add another tubulin so that that very, very terminal beta tubulin keeps the entire structure intact. Hold that off and the entire structure falls apart. So what does it mean for microtubules? They're like relationships. If they're not moving forward, they're moving backwards. You can never have a microtubule just sitting there doing nothing unless you treat it with Taxol. So in your cells, if you have a microtubule that's just hanging out and it's not doing a darn thing, it's going to depolymerize. The only microtubules in your cells then that are stable are the ones that are growing. You don't grow, you shrink. And again, the only exception to this are your neurons. In your neurons, microtubules don't undergo this process. They've got a different form of beta tubulin that has very, very little GTPase activity. So neurons are the outliers. The microtubules in your neurons are very stable. This is what the microtubules in all of your other cells are doing. Very dynamic, always changing. Okay? All right. Let's throw some other proteins into the mix now. So now you guys know how you can make and break microtubules. It's really that simple. How do we change their properties or structures? Well, now let's take a look at a neuron. Okay? So here's a typical neuron over here. We've got the cell body up near the top. Let's say this is a sensory neuron. Okay? Up here in the cell body, there's the nucleus. That's where the bulk of all the proteins are being made and assembled. We've got these dendritic progressions or protrusions up here. The dendrites are going to be the part of the neuron that perceives an external stimulus, like temperature. Okay? We work our way down the length of the cell, and here's the axon body. It's very, very large. It's very long. The axons in your neurons that are coming out of your spinal cord, going down your sciatic nerve to your big toe, that axon can be 3 meters in length. That's why you're going to need motor proteins to carry neurosecretory vesicles down the length of that. It's very long. We get down here, here's the axon terminus that's going to innervate, say, a muscle cell. So you stick your finger up here on a hot object, you pull it back because that stimulus is going to go down here and cause a muscle to contract. Here's the first dilemma you face in your neurons. The diameter of the axon constricts as we move from the cell body down to the axon terminus. The caliber of the axon shrinks. So if we take a look up here, near the cell body, here's all the microtubules. You have to space them out. And the reason you have to space out the microtubules, that's what vesicles are being trafficked up and down the length of the axon on. That's what the motor proteins are carrying those vesicles on. The problem is, you have to keep them far enough apart so the vesicles have room to move. So the cell here uses a spacer protein. And the spacer protein that it uses up here near the cell body is a microtubule associated protein called a MAP, M-A-P. One of the proteins it can use is MAP2. And MAP2 is a spacer. And what MAP2 does, it's got a region here that slips into the seam of a protofilament. And then what it does, MAP2 has this little sidearm that projects out perpendicular to the axis of a microtubule. And what it does, if we look up here in the cell body, the reason these microtubules are so nicely spaced apart is because MAP2 is shoving them apart. But notice what happens. As we get down near the axon terminus, the axon narrows. We've still got the same number of microtubules, but we have to bring them closer together. Well, we can't use MAP2 because it's too big. So as you move down the length of the axon towards the terminus, we replace it with another microtubule-associated protein that's called tau. Tau is much smaller. It's got a much shorter sidearm that comes out the side. And now what this does, it's holding the microtubules apart, but it's allowing them to pack somewhat more closely together. Tau can be a problem. And the reason is, tau can acquire missense mutations just like profilin does. And tau, when it acquires these missense mutations, it will aggregate. And these aggregation promoting mutations in tau are associated with Alzheimer's disease. And the reason is that the aggregating form of tau collapses the cytoskeleton as we move near the axon terminus. So in essence, what happens in these cells, among other things, is you can no longer maintain the proper spacing of your microtubules as we go down the terminus. What does that mean? Your microtubules all collapse down there. It brings vesicle trafficking to a stop. So your neurons can no longer send neurosecretory vesicles all the way down from their site of synthesis up here in the cell body and get them all the way down to fuse with the membrane at the synapse down there in the axon terminus. So it's going to have profound effects on vesicle trafficking. Okay? All right. So we'll stop it there, and when we pick it up on Friday, we'll actually go through some other modifying proteins, but that's when we'll move into looking at motors. Okay? All right.
That's a question.